This is the Redux News. Surplus of chromosomes may fuel tumor growth in some cancers. Without that extra genetic material, cancer cells form fewer tumors in mice. Some cancers are addicted to having extra chromosomes, a study in mice suggests. Cells usually just have 2 copies of each chromosome, one inherited from mom and one from dad. But about 90% of cancer cells have additional chromosomes, a condition called aneuploidy. Certain types of cancer cells often carry a 3rd copy of a particular chromosome or part of a chromosome. For instance, more than half of colorectal tumors have a surplus chromosome 13, and more than 40% carry an extra chromosome 7 or the long arm of chromosome 8. Stocking spare copies of chromosomes has been associated with poorer outcomes for patients compared with patients whose cancers have the usual 2 copies. It turns out that those extra doses of genetic material are necessary for the cancer cells to keep growing. Coming from a cancer geneticist, Jason Sheltzer, who reported December 11th at the joint annual meeting of the American Society for Cell Biology and the European Molecular Biology Organization. Put another way, cancer tumors are addicted to the bonus chromosomes, he says. The idea of addicted cancer cells isn't completely new. Scientists have known for decades that cancer cells can be addicted to altered versions of certain genes, meaning that those genes are required for the continued cancerous growth of the cells. As for chromosomes, researchers have speculated for more than a century that some cancers have particular chromosome surpluses that spur growth. But the ability to specifically delete specific chromosomes to test the idea is new, says Beth Weaver, a cancer cell biologist at the University of Wisconsin-Madison, who is not involved in the work. In the new research, Sheltzer of Cold Spring Harbor Laboratory in New York developed a method for purging extra copies of whole chromosomes or parts of chromosomes from cells. A type of ovarian cancer called A2780 carries an extra copy of the long arm of chromosome 1, known as 1q. Sheltzer used his manipulation technique to remove the extra copy of 1q from the cancer cells, then compared how well the original and 1q deprived cancer cells grew in lab dishes and when transplanted into mice. Cells with a surplus chromosome arm formed many large colonies in dishes and grew into tumors in mice. But cells that lost 1q barely grew at all, Sheltzer said. Quote, they've almost entirely lost their ability to exhibit malignant growth. Cells from which the extra arm had been removed later somehow regained another copy, restoring the cell's growth. These cells, for some reason, really want to have 3 copies of this chromosome arm, he said. That result is persuasive, says cancer cell biologist Adrian Soren of the University of Dundee in Scotland. Quote, that's a real sign of addiction. If you take it away and they manage to get it back again. End quote, he says. The idea that cancer cells can be addicted to genes forms the basis for many targeted cancer therapies, which interfere with the action of genes driving the cancer. Chromosomes, however, contain thousands of genes, so narrowing down which of those many genes or combination of genes is the source of addiction is much more complicated. But finding out which genes turn cancer cells into addicts is necessary if researchers are ever going to develop treatments to negate the effects of bonus chromosomes, Soren says. Quote, we probably need to understand a lot more of the biology of cancer cells before the new research becomes clinically useful, end quote, he says. He also adds, but I could see it in the future, end quote. Sheltzer took a step toward pinpointing why 1IQ has ovarian cancer cells hooked. The chromosome arm contains more than 1,000 genes, but Sheltzer found a likely culprit in the gene MDM4. That gene produces a protein that inhibits activity of p53, a protein that helps prevent cancer. With more MDM4 protein around, p53's tumor-suppressing activity is diminished, allowing cancer cells to grow unchecked, Sheltzer reasoned. To test the idea, he used the gene editor CRISPR-Cas9 to remove the MDM4 gene from the surplus 1q. Cells lacking the 3rd copy of MDM4 formed fewer colonies in lab dishes than cells with 3 copies, he found. But further experiments show that the gene isn't the only one spurring the growth. For now, the work is still preliminary, and Sheltzer hopes to do similar experiments with other types of cancers to determine whether aneuploidy addiction is common to all cancers. Thank you for watching, and let's hear what you guys think about this in the comments below, and don't forget to subscribe.